Hi, good morning, and welcome to the ZP webinar. That we like to do this webinar either once a week or twice a week, depending on the workload. But due to lots of questions this week, we've actually gone also on Sunday. So today is 3rd of December 2023. It's obviously Sunday. It's about approximately 7:30 a.m. and I'll just go straight into it because we've got quite a lot to cover this week. So don't forget, if you're looking for free information, there is the ZP Developer Zone. We do have courses up there that are free for people who want to understand electrochemistry. We do have the webinars. This is an example of a webinar. Today I'm going to specifically cover glucose and potassium sensing technologies. We do have collaborations. Now, collaborations, strictly speaking, by that term I really mean with academia. We do have commercial collaborations as well, but those have to be funded in some way because obviously everyone has to kind of, pay the salaries, cover the costs, etc. We do offer jobs through our LinkedIn, for example, is a good channel to find jobs upon from us. We do have our ZP Developer Zone, and we do have workshops. And it's probably worth saying that we're going to do a workshop next week, which is going to strongly cover, for example, potassium as a potentiometric or open circuit potential sensor. And we'll also cover glucose as well as a sort of example of amperometric enzyme sensor as well. So I'll talk about that as well. So there is in 2024, pretty soon we'll release a whole series of calendar events for 2024, but we do workshops almost monthly. I mean, if you look at this, you can see that we had 2 in January this year, 2 in February, maybe 3 in February, maybe 4 in February, 5 in February. So I can see that we do a lot of, this is the 2023, we have done a lot of workshops, and our workshops are not just talking. There's talking, but there's also going into the lab and doing experiments and making sensors as well. So we have a couple in December. If you see a link, by the way, I'll put the link underneath the video at the end as well. We also have free webinars. What's very specific or useful for today's webinar is actually our Sense It All webinar. So we are going to have a Sense It All webinar pretty soon. I think it's either the 13th or 14th of December. The webinars are free. Again, if there's see your link, then. But I hope that the questioners today will come to our Sense It All webinar. We're also doing a conference in the UK in June on the commercialization of electrochemical assays. So at ZP, we're super interested in, it's one thing to just kind of do some science in the lab, but you really want to get it onto the market in order to impact society. So we're super interested in that commercialization. And there will be a link here, and I'll put that link underneath as well. So if you're interested in the commercialization of electrochemical assays and biosensors, then please come to that conference as well. We also do on-site workshops. So for example, if somebody asks us to come and do a workshop with them or for them, we'll cover glucose sensors quite heavily. We'll in lots of formats, by the way, and the formats will come today in question number 2. I will actually mention all the different formats. We'll do testing of potassium sensors as an example of OCP, open circuit potential. I know the questioner today will use the term OCV, open circuit voltage, but it doesn't matter, potential voltage. They're pretty interminglable. And then we'll also do the theory of electrochemical sensing and things like, covering amperometry, voltammetry, potentiometry, impedance spectroscopy. Will also be covered, and we'll also do lots of demonstrations, how to do manufacturing, how to do what's called factory calibration. You'll see actually in question number 2 today about glucose sensors, how we do a really reproducible screen printed electrode, which really helps with factory settings and factory calibration rather. We'll discuss ISEs, ion selective electrodes, which again, the potassium sensor is an example of that. So we cover pH and potassium in that. Also discuss QMS, quality management systems. This will come up today in question number 2, but ZP is an ISO 13485 business, which means we're certified for contract development, contract manufacturing of, and we've just had our audit and it's of medical devices full stop these days. And we're also pretty good at project management. The philosophy at ZP, whether we're having a conversation about glucose sensors or potassium sensors, is our philosophy, and we're pretty good at this, is actually to get these kind of sensors into people's hands really quickly. So on one left-hand side, you'll have kind of engineers and scientists and entrepreneurs and businesses. And on the right-hand side, you'll have the kind of end user. And we're really good at kind of packaging technology. We have technology already packaged up, by the way, in something we call the Sense It All platform, which we can marry with any of our sensors, for example, the potassium sensor or the glucose sensor. So that package is essentially ready to go. And then you can start putting it into the hands of the people who you're really expecting to use this. And then because we make everything cloud enabled, so if you're using one of our Kate potentiostats that'll come up today, or one of our Sense It All platforms, they're all cloud enabled. So the data will immediately go to the cloud, which means you can start kind of looking at that raw signal. And if you need to kind of make improvements to make it more accurate or precise for the end user, you can do that through the cloud. So we're very keen on not spending years developing, but actually spend some time developing and get the technology into people's hands, and then really sort of getting that feedback. I mean, this is really a kind of a lean startup kind of mentality. So at ZP, you'll hear us talk a lot about MVP, minimally viable products, and you also and these kind of philosophies of being quick and sprint sometimes is a good term. I mean, these are really lean startup mentalities. I will jump into the questions in a minute, but many of our sensors are compatible with this philosophy, or are compatible. Like we've got the glucose sensors today and the potassium sensor, which I will zoom in on. So we are going to cover those two sensors. There we are, glucose and potassium. What's quite interesting about actually those two analytes, when I think about it, is they're both unfortunately associated with kind of chronic diseases, whereas glucose is often associated with diabetes. Potassium is also an indicator of the treatment regime necessary for dialysis patients. They're both chronic diseases. So they have, unfortunately, and I do mean this, that there are some, inherently business models built into them that the people will require continuous or frequent testing. It doesn't have to be continuous, it's just frequent. So for example, a diabetic can test 4 times a day, or at least they're asked to do, or they can wear a CGM, or continuous glucose monitoring, but it doesn't have to be continuous. You can start off with frequent discrete testing. So we do have these Sense It All platforms. This is the spectrochemical one. So if an assay is spectrochemical, if it's electrochemical, this is our electrochemical point of care or IVD platform. And we also have a strong wearables platform as well, because I've spent a decade and a half doing continuous glucose monitoring, but we're also very strong on lactate and ketone, and we hold something like 50 patents on wearable biosensors. Something that underpins everything we do at ZP is probably manufacturing, and one example of manufacturing is our screen printed electrodes. So one of the questions that's going to come up today is they are going to ask about platinum electrodes for glucose sensing. So we do have some really good platinum sensors, and this will jump up in question number 2 today. What makes these unique is the quality, but that quality is actually reproducibility. The biggest problem with, for example, screen printed electrodes that are put into the R&D market is reproducibility. Big difference in ZP is we are the biggest users of our screen printed electrodes, which means that we need the quality and reproducibility of those electrodes for ourselves, and therefore it helps our clients who also use them. But we're not just producing them for our clients, we are actually using them ourselves. I think the analogy is we are essentially chefs or cooks that eat in our own kitchen or our own restaurant. So we know if the food is bad before anyone else knows. So I'll put some links to those screen printed electrodes, but it will appear in question number 2 about the platinum ones as well. So question number 1 is really about potassium sensing. So I should have put a big title here which says potassium sensing. We work with potassium sensors obviously on our bench, and question number 2 will be about glucose strip technology. What technologies does ZP have around glucose strip? So question number 1, they want to check the functionality of OCV, open circuit voltage, with a research potentiostat. It's a very small thing, by the way, but potential is the sort of name of the property, and voltage is the unit. So I know people use OCV. I get criticized by electrochemists sometimes, though I am an electrochemist, by the term OCV, because they say, oh, it's potential and the units are voltage. I think it's very pedantic and I don't care, but just out of interest, you might get people picking you up on this term OCV, and you'll see in software often it's of OCP anyway, but don't worry about it. So they want to check the functionality of potassium sensors using OCV. They want to connect the sensors afterwards to an analog front end. I'll talk about that. They have got ZP potassium sensors and solutions, and I will discuss that. They have been reviewing our online material for setup. If people get stuck and they are users of our technology, we will make videos, etc., to try and support them. So just know that. This is an interesting one. See, they want to target the open circuit voltage in the range 150 to 200 millivolts. This one I found quite curious. If you come to our workshops, we're not going to be pedantic and derive the Nernst equation, but there is something called the Nernst equation that kind of says if you've got a concentration change, let's say you went from 4 to 14 millimole of potassium, theoretically you're expecting a kind of potential change there in that of 59 millivolts. Yeah, that's about right. 59 millivolts. So I'm a little bit sort of, I won't say confused, but this 150 to 250 millivolts, your signal is, its range is going to be greater than this 50 millivolts is my suspicion, but I'll give you some data to kind of cover that. They're talking here about PBS, phosphate buffer solution. I mean, our phosphate buffer solutions are generally kind of, generally we have a kind of saline concentration or a sodium chloride concentration there of about 150 millimolar. We buff them at about pH 7.2, and we make the potassium at their concentration indicated. So so they've bought the kit off us. So I think the potassium concentrations that we give in there are 0, 2, 4, 6, and 8 millimolar. But I'll cover that in some slides in a minute. Ask a recommendation on the concentration of PBS used to coat the working electrode with desired OCV. Yeah, it's kind of interesting. See, it's quite a discussion actually, because you can tailor theoretically you can tailor a PBS solution to give you on that working electrode a potential about 150 to 200 millivolts. At that point though, it's not PBS. See, the PBS phosphate buffer, I think the S actually, I sometimes say solution, I actually think it means saline. It's meant to be mimicking the blood. So everything, all the electrolytes are kind of approximately what would otherwise be in the blood. So you can change the phosphate buffer solution / saline solution to give you potentials maybe of 150 to 200 millivolts with that potassium sensor. At which point then it's no longer saline, it's not a blood mimic because it doesn't have the same electrolyte concentration. So I find that a little bit curious. They are interested in running sensors over extended periods and observing drift. So I'll show you some data of 1,000 seconds today, which I know you'll want longer, but 1,000 seconds is 20 minutes approximately. And they want to seek for advice on prolonged use. Now, ZP, our business model is we do contract development, contract manufacturing. We have standard products. The standard products are intended really so people can go from 0 to 1 really quickly. In the bad old days, in order to go from 0 to 1, you'd have to go away and build an entire potassium sensor, then, and that would get you from 0 to 1. But at which point you'd find out that it didn't work in the first instance and you're back to 0. We've kind of solved that problem. But we do also then consult as well. So we will give private support on the sensors, especially for people who are kind of seriously wanting to commercialize something. Question number 1A, they want to use it with a potentiostat. Now, I know the brand of potentiostat that they have. And that potentiostat that you have, if you look at the cables, you've got banana plugs on the cables. They're probably 2 millimeter in diameter, and our sensors are 7 millimeters in diameter. So there's a bit of a mismatch between your cables and our sensors. So you actually need a connector. So we have connectors which have 2 millimeter slots at the back for the banana plugs and a 7 millimeter slot at the front for our sensors, and I'll put a link to that as well. So my biggest, let's say, little concern at the moment is you may not have connectors to go between your potentiostat, which you told me. I'm not going to discuss it in the webinar because it's told in confidence. But I think you actually need a connector, and I'll put a link to that connector here. If you've got poor connection, because you're doing open circuit voltage, that's what we call, a high impedance measurement, a high resistance measurement. So it's probably less important, but when you're trying to do other techniques, if you don't have good connection, you can end up with quite sort of, I won't say corrupted data, but data that's not so so good as it should be, because otherwise it's actually the high resistance connections that are the problem. So we came up with these connectors. Actually, it was on a potentiostat that you've got as well, that somebody had poor connection between their cable and our connector, and we had to end up, our sensor. We ended up having to build them a connector, so that's why it came about. Question number 1A as well. One thing I would say about the potentiostat that you have is it'll be very frustrating after a while that you're actually only able to get one data point at a time. So this is more for our sort of medium-term thought as well, is actually you will need to get parallel potentiostats. Be careful, don't get multiplex potentiostats. Multiplex potentiostats, I don't want to say I hate them, but they kind of miss the point a little bit. You get all these sensors in the front of it, but because you're trying to save cost, you kind of go, the electronics just get switched from one electrode to the next, or one sensor to the next, and I think it really, it doesn't save time in the long run. I really like true parallel. You get all the data at the same time, and it really saves cost. So we have something called EMMA. It's a true 6-channel potentiostat. Be careful because it's not multiplexing, it is parallel, which is, I think, is a really important point that we get. And I suppose at ZP, because we do a lot of this work, we do everything in parallel practically, because time is money essentially, and we don't want to make business decisions based on really small sets of data, so you need to go parallel pretty quick. Sorry, and if there's a link, I've put a link underneath the video, or at least I will put a video. These people don't have the CAPE potentiostat. I'm just going to bring up the CAPE potentiostat, because the CAPE potentiostat is a real, it's a lab instrument which works really well with our screen printed electrodes and our biosensors. But it's also our Sense It All platform. So this means that people can go from kind of lab experiments to actually minimally viable product, or even product, because it's actually so well manufactured. And it's part of our lean manufacture, our lean startup mentality, that your lab equipment is also actually your products, and it really kind of breaks down or accelerates the time to get to market. And I'll put a quick link to that as well. So these people are not using Kate, the potentiostat, they're not using the Sense It All platform, but for other people who may be following on later on, we do have a very nice R&D solution, which is also, the elegance of it also makes it your product as well. So suddenly this kind of disconnect between what you did in the lab and what you would give to the first alpha and beta customers, it's not a different unit, it's the same technology. So I'll put a couple of links to that as well. Sense It All, I am asking both of the people who are asking questions about the glucose and the potassium sensor to come to a webinar that we have on the 13th of December. But I'm realizing that I'm actually going to ask the people who are asking about potassium to come to a workshop, which I think is not next week, but the week after. It's about the 12th of December, I believe, in Coventry, and I really hope you come to that, because actually we could do some potassium sensing there as well. Anyway, there's a webinar. Both questioners this week, please come to this webinar. It'll be really useful and enlightening for you. I'm going to get back to the point in a minute, but the CAPE potentiostat, the Sense It All platform, work with all of our sensors, and there's a link here as well. Small matter, but this will come back later on. If you've got an Android smartphone, at some point rewind this webinar and just scan this, because this is our Sense It All platform. So what we're really good at ZP is deployment, getting things out of the lab into beta alpha testers, or even in front of investors really quickly. This is called the Sense It All. If you scan that with a QR reader on your smartphone, especially Android, it will allow you to install something called Sense It All, and I'll come back to that in a bit. So Sense It All is a way of going from an idea to a minimally viable product really quickly. We have a particular set of electronics in there, and I'll discuss it here. So for example, if I'm talking about glucose or potassium, what we have is glucose and potassium sensors. We have the Sense It All hardware. We have the app, which I just gave you a QR code on. And we have something called Julie, and I will discuss Julie in a minute. Julie is something like getting close to 10 years old at this point. It's something that we've put a lot of, and it's on its second major iteration in terms of, we've kind of saved all the good features but scrapped the code base and started again, which is often something that happens in tech development. But Julie is a cloud database system where the QR codes that I was discussing earlier on interact. It's where we store all our data. So it's a cloud data management system, and I'll give you some more details about that. But sensor goes into the hardware, for example. Sample goes onto the sensor. It could be a dialysis sample for a dialysis patient, or it could be a glucose sample from a diabetic patient. The app tells the hardware what to do. The hardware gives the data back to the app, and the app sends the data onto Julie, and you get some sort of concentration, millimolar, ppm, pH, depending on what you're actually measuring. But Julie also puts the app also puts the data into Julie. So if you're doing some sort of lab testing in a lab somewhere, if your colleagues are asking you where the data is, don't worry, it's all on its way up to Julie. Or more importantly, or let's say importantly, if you're working with off-site people, the question is, where's the data? Don't worry, the data's all in Julie. Or if you're working with trials with patients or preclinical trials and it's remote work, don't worry, the data's all going to Julie. And that's really powerful as well then, because it means that people like ZP can actually look at the data with you as well. As long as you share it with us, the optimum, as long as you want to share it with us, we can see the data then as well and advise. So SensiAll is a really nice piece of hardware. It's got the kind of LEDs which guide the user experience. Is it on? It turns white. Is it made Bluetooth connection? It's blue. Is it running the assay? It's flashing green. Has it uploaded the data? It goes solid green. So a really kind of nice user experience built in just on the LEDs, let alone the mechanicals of this as well. Just showing a few different angles of it. Like I say, see, the nice thing about it is the SensiAll hardware that we're using here, it's also the Kate potentiostat. So it's a kind of almost like a consumer electronics experience in the lab and something that you can get back out of the lab. In one of the videos here, I'm just showing the kind of hardware. The fact is it's got this very elegant way, and I have said that there's that webinar on the 13th of December. Please sign up for it. The hardware, you take the sensor out quite easily. It's not trying to, a problem is real people don't want to be slotting things in and out of horrible slots, and that happens with a lot of sort of lab potentiostats. You can see here that we've got a nice leverage clicking system. If for some reason it gets dirty, for example here, we use this sensor for sometimes we use the system sometimes for measuring soil, for example, and so you get some soil on that, we can wipe it down and clean it. We use it for saliva testing. You can if you get some saliva on it, you can just dismantle it and clean it, put it back together. It's not just the hardware, it's actually part of an entire system where we've got the app, the SensiAll app, which resides on the Android Play Store or the Google Play Store. That app makes Bluetooth connection. Come to our webinar, you'll see us do this kind of testing. And when they've completed the testing, as I say, all the data's in the cloud. And then elegantly, we can go through between different analytes. We can reskin the entire app and go to different method files, different analytes, just by using QR codes as well. And I'll bring up some QR codes. Oh, in fact, here we are. So, for example, if you've downloaded the SensiAll platform and created an account on Julie, you can scan these different QR codes, and it'll just give you a quick example of how you can go from chili to ginger to nitrate to total antioxidative status. 4 quite different analytes, 4 quite different applications, but it just gives you that term SensiAll kind of means something. This is Julie itself. So Julie is the back end of the SensiAll platform. So we have data storage, data visualization, which is really important, data processing. We can do filters, we could do online calibration, and then obviously data acquisition, which Kate is the potentiostat for the lab. SensiAll is really Kate, but for more for application focusing, more what you would give a customer experience with. And then it also has API built in as well, application program interface. So if you're a company that's developing sort of, let's say, deep tech, it could be just a piece of silicon to go into biosensor applications. Understand that getting into this platform actually brings what you're doing or data that you're gathering all the way to the cloud, and it opens up a whole new sort of business model ecosystem for you as well. And what's really nice about Julie then is it's cloud-based. You can share data in that. You can create groups, and this is what's really powerful for ZP as well. That I can be in the UK, the team could be working in Norway, and I can see their data quite readily. In fact, I've got customers in India, and I've really been able to analyze their data and explain to them that what they're seeing is real, and give them confidence that the data is right because of this really network effect, which is get the data to the cloud. The more people that look at the data, the more value we can actually add to the data. So we do have a real network effect there. Analog front end, these people are very interested in on the potassium sensor transitioning to their own analog front end. It's probably worth saying that at ZP we are quite used to working with semiconductor manufacturers. We're not a semiconductor manufacturer ourselves, but we do have we do the sort of we come up the value chain in terms of we take semiconductor components, analog front ends, we put them into these chips. This is something we call the single purpose biosensor circuit, and the circuit that we have here in the hand of this user here in the left hand here, and she's just putting it onto that breakout board. It's something we sell. It's something that's at the heart of everything we do. So sensors and this chip are sort of essentially, we're keeping consistent with that chip. So just to know that we are quite used to working with semiconductor manufacturers and properly shielding it and essentially giving these pieces of silicon application through biosensors. Something that we do have then is, for any of our sensors, we can essentially create a QR code which can be read with Sense All. So even if the electronics change, a new piece of silicon comes into the ecosystem, we can, rather than you having to tell somebody how to program that chip for a particular analyte, ZP can create QR codes which are available and which can be read through the Sense All platform, and you can end up programming the chip essentially in the field for the particular analyte that you're interested in. To just know that ZP has created a workflow for developers and manufacturers of analog front ends. So rather than them then having to determine how a customer is going to, or what parameters, what potential, what current range are they expecting, rather than that all being programmed in, you can actually give the user experience a kind of almost like a QR experience. So it really helps onboarding of new clients. So back to this, they do have our potassium sensors. I'm super proud of our potassium sensors. They're one of the oldest sensors we've got, and that really means that they've had tens of thousands of ZP effort got into them actually over the years. They also have the calibration solution from us as well. Just on the calibration solution, the solutions they bought, but they have 0 millimolar, 2 millimolar, 4 millimolar, 6 millimolar, 8 millimolar. If you come on our course, our workshop, I will laugh about the 0 millimolar. The reason being is there's an equation behind how a OCV or open circuit voltage or open circuit potential or ion selective electrode sensor works. There's an equation behind it which is the Nernst equation, which kind of says the signal is proportional to the log of the concentration. The log of 0 is infinity, so it's a kind of it sort of breaks the equation that we have behind it, but it does work. But I think it's better with potassium to actually not test at 0, test at 0.1, but you'll see our data, you'll see that it is working. Don't worry about it. I'm kind of curious about this range that they want, this 150 to 200 millivolts, because if I look at our raw data, this is our raw data off the website, by the way. Yeah, and I have a link for it. Good. I can see in this data that we've gone from 4 millimolar to 16 millimolar, which is approximately 10 millimolar. So we've got about 55 millivolts range. We've gone back from about, I've just done this approximately this morning. I think my maths is a bit worse than I was actually expecting. We've gone from about 350 approximately, is that right? That's 315. We've gone from 315 millivolts to about 370 millivolts, which I think my maths is about 55 millivolts over that range. Theoretically, we should go about 59 millivolts per 10 millimolar. So we're close to a theory, but not absolutely on it. But to be honest with you, it doesn't matter to be absolutely on theory. We got enough signal. You can see the signal, in fact. I mean, 4, 5, 6, 7, 8, 7, 7, 16, 8, 6. So it gives you a sense actually of our, and this is quite interesting actually, because when I look at these signals, even though we're changing concentration coming back again, we're getting pretty close to what we had before. So this slightly answers the drift question that will come later on. But the first question is, see this 150 to 200 millivolts. Well, the first thing is, you can see that, in fact, the potentials we're getting and then the solutions you're getting, you should be getting potentials of approximately 310 millivolts to 370 millivolts. It's not that. And the range is greater than 50 millivolts. So that I find a little bit curious. Question F. It says here, concentration of PBS on the sensor to achieve desired voltage. If you want 150 to 200 millivolts, we could probably make you up a, I want to say, bespoke solution to give you that potential range. But then it won't be PBS. It'll be a phosphate buffer, but it won't be saline. It'll be something else. So we could do it, but I'm really not sure that the question is the right question, or what you're trying to do is quite right. Just out of interest, if you want to get data like this, then buy our solutions. You have bought our solutions, and therefore you are actually going to get, you should get data pretty similar to this, and the solutions you'll get are 0.2468 millimolar of potassium in those solutions. They're interested in drift long term. Just out of interest, what I'm looking at here is about 1,000 seconds worth of data. You can see a bit of drifting around here. This is because we're at 0 millimolar. In most samples, in most clinical samples, you're not at 0 millimolar. You're at something like 0.01 millimolar or 0.1 millimolar. It's just us. It's just a historical accident. But that said, so ignore the 0. Then they had 2.7 millimolar. When I look at this, I can see let's look at each one in line. A little bit of drift there. This one comes up and plateaus. This one is solid. This one's solid. This one's solid. This one's solid. A little bit of a drift downwards there. A little bit of a drift downwards there. Then they come to 4 millimolar. Pretty solid, pretty solid, pretty solid, pretty solid. In fact, they're all solid, because at this point now, the sensors are probably kind of equilibrating, let's say. At which point, some of these sensors I can see drift in them, but some of them are actually solid as crazy. So at ZP, we make the potassium sensors available to people so they can go from 0 to 1 really quickly. If you want to go from 1 to 10, let's say, then that's a different conversation. But I can see just on the sensors that we make available to the R&D community, actually some of them are pretty stable. If you don't have stability, it's probably your experimental setup. One of the biggest things, one of the simplest things is people put solutions on, or they put the sensor in a solution. Solution starts evaporating, the ion concentration changes, and they start getting drift. But it's because the solution's not stable, not because the sensors are not stable. Just out of interest on this one, they wanted to put it in a strip format. A strip format is pretty good. It's a really good way of manufacturing. It's very scalable. I'm also going to talk about strips in question number 2 as well. So hang on for question number 2 as well in a minute as well, if you don't mind. So let me go into question number 2. And I'm sorry that I think that entire conversation might have just been about 40 minutes. 40 minutes on potassium. So please come to one of the workshops. You can see that it's almost a day's conversation in itself. All right, question number 2. Let's get on to screen. So these people want to make a glucose sensor using the catalytic properties of something like platinum. So ZP, we do have, one of the things you've got to be really careful about is people really jump into having bespoke electrodes far too early. Before you start going on the customization of electrodes, get some samples, or you can buy some samples from ZP of our materials. Just make sure that you like these materials before you start going down the route of customization. I suppose, again, it's that lean startup mentality. Do things that are quick and low cost relatively. Customization is not quick and it's not low cost. Don't go there straight away. So we have a slightly side point. If we're contracted to be consultants on programs, we will talk about the relative benefits of carbon, gold, and platinum. But today, anyway, I've got a whole list of different carbons and different platinums and the gold electrodes. These people are particularly interested in platinum, so we have the 501 platinum, and we also have the hypervalue platinum as well. And I've put some links to that's the 501 carbon. This is the 501 platinum. Not this carbon, but that's the hypervalue platinum that I recommend, and that's a gold one there as well. So I recommend of the previous slide, the 501 and that hypervalue platinum, and I'll put links underneath. So if you're trying to build a glucose sensor, particularly on platinum, these are the two screen printed electrodes for you. I'll put the link underneath. I'm not going to go back through the whole Sense It All and Kate's conversation, but if you've jumped forward just to go to question number 2, just know that ZP does have platinum-based glucose sensors already, and actually you can really just sort of jump, I would say, sort of 2 years worth of effort by just starting to adopt our Sense It All platform. 2 years is just an estimate, by the way. I mean, we've been working on this for, we're in our 10th year at this point. So that's the kind of, and hundreds of thousands of man hours have gone into glucose sensing, no doubt about it. So question number 2, go back through question number 1 and look at the whole technology stack if you don't mind. It'll really save you a lot of time. Because we're talking about glucose strip, and the last question on potassium actually finished a glucose strip as well. I just want to kind of say that at ZP, we're really good at making reproducible screen printed platinum sensors. I've recommended 2 of them. By far for the R&D market, they're the world's best. We're also good at creating microfluidic channels as well. These are some examples. This is a slightly bigger microfluidic channel, but it does show it better actually, that whether you're making a potassium strip for a dialysis patient or you're making a glucose strip for a diabetic, we can basically use the same technology that fills within, 1 or 2 seconds, and we're really good at making these kind of low cost microfluidic channels on top of sensors. Something that's really important about ZP's screen printed electrodes, if you come to our webinar, which I've asked you to sign up for at the very beginning of this, you'll see that when we're doing demonstrations, we won't do any calibration. And the reason that we don't do any calibration is really the reproducibility of the underlying screen printed electrodes at ZP. So at ZP, our mentality is we're trying to do everything calibration free. If you're bringing calibration to the end user, it's because you probably don't have control of your manufacturing process. I mean, it happens all the time in academia. They either normalize the data or they have a calibration step, and it's really because the electrodes they're buying in or the sensors they're building are not reproducible. You'll find with us. So what we're really good at is we actually we test the electrodes before we either use them ourselves or before we use them with clients or before we ship them to clients. So we will reject this sheet. Sheet 1 always gets rejected, but then we'll be checking them. And we're the only company that's checked it by one of the companies that does it the best. We check by electrochemical means that these sensors are actually going to function the way we're expecting. It's a big effort and therefore a big cost to actually do all this testing of reproducibility. But it means that our coefficient of variation or our relative standard deviations are single digits. So here I'm looking at several sheets through a manufacturing run, and I can see the mean is 5.8, the mean is 5.8, the mean is 5.98. So you could call that essentially 6, 5.8. So here's our means, here's our standard deviations, here's our relative standard deviations, and then we track it over the entire batch as well. It's this data collection around manufacturing that makes us, it's the manufacturing itself, but it's also the way we do the data. So that means that when we're doing something like signal versus concentration, we've got three sensors here, 3 sensors here, 3, 3, 3. We can get very good signal versus concentration, and we can we don't get overlap between different concentrations because of the underlying reproducibility of the electrode, and therefore we can be calibration free in our applications. But I think what happens with screen printed electrodes a lot is people just go rushing out, can I have free samples? That's always a question, which I think is, it's not a good start of a business relationship to ask for free samples. And also then they're going around looking for cost, but cost you'll end up paying for. When if you're looking for the lowest price, and you will get the lowest price, but you also get the lowest quality. I've come across one company where they said to us, ZP, you're 3 times as more expensive than another company. They went with the other company. In the end, they found out that the yield was 30%. So essentially, they paid for the difference in our cost and the other company's cost through yield. So they end up paying the same amount, but then they had to deal with all the filtering of that material to actually get to find the 1/3 of the electrodes are actually any good. So just as a background on ZP when it comes to CGM or glucose monitoring, it's probably worth saying I started my first glucose strip engineering job in 2000, so I've been doing this quite a long time. We launched ZP in glucose sensing. We've been doing glucose sensing in this business since 2014. We are ISO 13485. We are contract developers and contract manufacturers of electrochemical biosensors. It is important that this is a privately owned company. We're not VC funded, so therefore it is important that there is a budget in place to kind of, cover ZP. We have 4,000 square meter facilities, so we're not big, we're not small. We have facilities in the UK and Norway. And as I say, here's a couple of pictures of. I suppose it's worth saying that our facilities are, state of the art in terms of clean. We're doing clean room. Our R&D takes place in a clean room, and our manufacture also takes place in a different clean room. So we got a lot of capabilities when it comes to glucose monitoring. We do wire sensors, microneedles, planar electrodes. We do the characterization and testing. We do the electronics. We do the rigs and jigs. We can do multiple analytes on one sensor. We do documentation in accordance with ISO 13485. We also have what's called aseptic manufacturing, i.e. clear from bacteria. This is some of our wire sensors. These ones get implanted into people. We spent a lot of time and money making this sensor. This sensor actually gets implanted into fish. So that's sort of talks about the robustness of what we actually get up to at ZP, that we can put a glucose sensor into a fish and leave it there long term. On microneedles is another sort of popular way of doing glucose sensing these days. ZP has 50 patents around this. And it's not just, these are manufactured in a sort of aseptic environment. They're properly packaged. So we really know how to be kind of professional around this. We do have a lot of microneedle experience. I don't think I've kind of I've kind of left out the bit where actually we also do a lot of planar electrodes. So the whole conversation here is about screen printed platinum electrodes. We do it on screen printed platinum electrodes as well. We're really good at testing. We're really good at interference testing, because we're an unusual company that we have electronics, applications, domain knowledge, and also we're wet chemists. So we have biologists, material scientists, electrochemists, chemists. We have a lot of the wet science disciplines covered. It's not enough just to make these kind of things. You have to make lots of jigs and rigs in order to test these kind of sensors. So we've also got a strong electromechanical team as well, just to help us make the test jigs necessary to do this. It's not just glucose. We're also able to kind of do this kind of work. For example, we can do a glucose and ketone sensor at the same time. And we're actually testing glucose and ketone at the same time and showing that, what the effect is on interferences. We are ISO 13485, so we do follow, sort of, if brought properly into a program and it's properly funded, then we're able to kind of do the definitions part of this, ideation. We go through the entire kind of design history file or design history record, making sure that everything is documented. People don't always like documentation because they're paying for something that they're going to benefit from later on, whereas they want kind of tangibles. They want you to go quick, but then they kind of regret it later on because later on you do need this documentation, to sort of, the FDA will expect at some point to kind of have evidence of all of this, and so we're pretty good at doing the quality system as we go along. As I say, we do have aseptic manufacturing. This is genuinely us doing glucose testing in our lab, and it's no joke, that we are wearing these kind of bunny suits, PPE, to make sure that we protect the devices from us, let alone us from things that we're testing. It's actually the other way around. We do a lot of CGM or glucose manufacturing. We do planar wire, microneedles. So I think I would sort of finish it like that, and I'll do a quick summary, let's say. So on the potassium sensor, you've got our solutions, you've got our potassium sensors. I think the thing you might need is actually the connector, at which point you should be in a pretty good state to do open circuit voltage measurements. The workshops could be super interesting to you if you get stuck. If you get stuck as well, if you were on our Kate platform or a Sense It All, it would be better, because then I could actually support you in real time. As you do an experiment, you could share the data with us. We could look at it, but you're not, but that's okay. And then on the glucose front, I would rewind this video a few times, but it's probably, if I was to summarize it, ZP at least I'm one of the principals. I've got 23 years of industrial experience on glucose strip manufacture. We do glucose sensor manufacturing all of the time. And we have kind of a deep knowledge because it's not just making glucose strips. We also have continuous glucose monitoring experience as well. And then it's kind of interesting because we have the kind of microfluidics experience. We also have the electronics experience. We're quite used to measuring glucose in low glucose regimes. So for example, interstitial fluid is not the same concentration necessarily of blood. And I've done sort of microneedle work as well, which is actually, you can really, and we've also got quite a lot of experience on analyzing analytes in sweat. But in that case, it's actually lactate, but we are used to monitoring different analytes. So I think I would wrap it up and say, if you've been watching along this webinar, please don't hesitate to reach out to us. If you've asked question number 1 or 2, please come to that free webinar. If you're question number 1, there's also a workshop in a couple of weeks in the UK, which would be good if we can get you down for that. So in summary, if you've got any questions, please don't hesitate to reach out to us. Okay, thanks very much.